Computation. Oh, I have to click. Yes. Okay. Okay. So the motivation of this work is that I wanted to model an organism as opposed to modeling the responses of some neurons to stimuli, but really modeling an organism that would behave in an environment. And of course, the difficulty is that usually organisms are very complicated. So if I wanted to model a mouse, for example, that would be quite challenging. Uh, so some people, for example, have got interested in simpler organisms, simpler species like C. elegans, for example, which has about 300 neurons. But that is actually still very complicated. So I was quite happy when I stumbled on this little thing I'm going to show you, which is Paramecium. It's a unicellular organism, and it has some features of neurons. So let me show you what it looks like. So this is Paramecium. So there are several species. This one is Caudatum. It's about 250 micrometer long, so it's a big cell. It's a single cell, but it's a big cell. And it's covered with cilia. As you can see here, it has a few thousand cilia. It beats its cilia, making waves, and it swims in fresh water, in ponds, in lakes, all over the world. And it has been doing so for several 100 million years. So it's a very old species. What you see on the right, for example, is a fossilized paramecium found in Triassic amber, more than 200 million years old. Um, so, um, so it's an old species, basically. Uh, Sorry. Yeah. Okay. So here you see some paramecium that is swimming in a petri dish. So typically it swims in a helicoidal path. And then you see that sometimes it changes direction. And this change in direction is called the avoiding reaction. Oh, sorry, I have some problem with the PowerPoint. Yes. So this is called the avoiding reaction. And this happens either spontaneously. You can see two spontaneous avoiding reactions here. Or it can happen when there's a stimulus, for example, a mechanical stimulus. So here when it touches the edge of the petri dish. And if you look closely, what you see is that it not only changes direction, but it actually slightly, for a brief moment, it swings backward, and then it turns around itself and swings forward again. This kind of stereotypical behavior of paramecium that was observed actually more than 100 years ago, and particularly by Jennings. And Jennings wrote a fantastic book about so-called lower organisms like amoeba and paramecium and all that, where he describes the behavior of these little things. Uh, so the reason why I got interested in paramecium is that this avoiding reaction is triggered by an action potential. So a bit like in neurons, except it's a calcium-based action potential. It's also graded, so it's not exactly the same, but it's still um, the same biophysics, basically. And so it's an excitable cell. What you see here is an experiment that was done in the 70s. There was a lot of electrophysiology in the 70s in paramecium. Um, People actually at this time were interested in doing electrophysiology in lots of different species, invertebrates, and also protists like paramecium. And so they realized that paramecium produces action potential. So here they sent a short current pulse. And when the pulse is strong enough, you get an action potential. And this action potential correlates with the reversal of the cilia. So during the action potential, the cilia reverse, and so the cell swings backward. And then it swings forward again. And this happens because this is a calcium-based action potential. So during the action potential, the calcium enters into the cilia And so it binds to different things in the cilia, and that makes the cilia direction revert for a brief time until calcium is expelled. So at this period in the 70s and 80s, a few people working on this were, uh, they call this paramecium the swimming neuron. It's a little bit like a neuron, even at, in fact, at the molecular level there are, and biophysical level, there are many similarities, as you'll see. So today I will, so in this talk I will talk about two things. First, I want to show you a little bit about the behavior, what this thing does as an organism. So this you will find in a review that I wrote, that was published last year in Neuro, where I discuss paramecium, but from a neuroscience point of view, so behavior electrophysiology. And then in the second part of the talk, I will show you well, the work we've done on modeling the electrophysiology and the behavior of paramecium. So as I mentioned, um, there's an amazing book from 1906 by Jennings called The Behavior of the Lower Organisms. Well, there are lots of details about the behavior of paramecium and also a few other species. So many things that I will show you are actually coming from this book. So, sorry, the camera gets in the way of, but it's okay. Um, okay, so on the left, you see an immobilized paramecium against the filter. And so you can see the cilia beating. They beat very fast, like 20 hertz. And that produces a wave. It's called a metachronal wave on the body. And this wave makes the cell move forward in a spiraling movement. Like many microorganisms, actually, it swims in spirals at a pretty large speed, up to a millimeter per second, which is roughly 10 times its body length per second. So it's quite fast. It also does gravitaxis. It tends to go to the top of a tube, for example. Not exactly clear how it does that, actually. Um, then, and sometimes it attaches to fibers or some other things, and then it can feed. So here you see an example of paramecia that are feeding in their culture medium. And in fact, yeah, you see a little inset is the experimental setup. This is something I did during the first lockdown here in Paris. That's my setup. It's just with a simple USB camera. So that's one experimental advantage of Paramecium. It's pretty simple to culture and also to manipulate, to do experiments. It's pretty easy to do it even at home, actually. Uh, so that's one of the advantages. All right, and then it's also mechanically sensitive. So here you see paramecia that are bumping against actually a piece of my jumper. So you have fibers here, and if you pay attention, you will see avoiding reactions against those fibers. So it's very sensitive mechanically, as you can see. Um, so it does those avoiding reaction. It can also kind of jump. So this is the second video here. It's in slow motion here. What you see on the video on the right is a case of jumping by throwing little spears called trichocysts. So here the stimulus is a laser which heats basically the fluid near the cell. And when the laser is on, it's going to launch, yeah, trichocysts toward the source of the heat and jump laterally like that. Um, so that these are a few examples, and then it's sensitive to many different modalities. So I've mentioned the mechanical sensitivity, but it's also sensitive to many different chemical substances. For example, it likes weak acids. So here you can see it's actually a drawing made by Jennings more than a century ago. He puts a little drop of acid and the paramecia go inside and gather inside. And you can see that they kind of bounce against the boundaries of the drop, the acid drop. They are also thermally sensitive, so they tend to aggregate at a temperature which is close to the temperature they have been cultured in. So in fact, it's a case of thermal regulation by moving. They are also sensitive to light, to bright light. So they don't like bright light, so they tend to gather in the dark. And there's also some collective behavior. So for example, here you see a group of paramecia that get just gather in the absence of any particular stimulus, and that happens because they breathe, so they produce carbon dioxide, and that makes the fluid a little bit acid, and they like acidity, so there's a collective behavior there. They tend together. Um, so generally, the logic of Paramission behavior is pretty simple. It's an exploratory logic. Basically, it swims in kind of in a straight fashion, well, a little bit in spirals, but they swim forward. And when there is something that is not desirable that they don't like, they change direction. So, and by doing so, they can do lots of different things. As you have seen, they can do thermal regulation, they can gather in acid places, they can do chemotaxis, find bacteria. Yeah, I didn't mention that Paramecia, they feed on bacteria mainly and yeast and a bunch of other things. So they can locate them, feed on them. And so they when they do the avoiding reaction, they change direction randomly, or at least it looks random. In fact, it's rather pseudo-random. It's just that they always turn in the same direction, but because they spin at the same time, it ends up being in a different direction every time. So in addition to that, there's also adaptation. That is to say that when the medium is changed or when the temperature is changed, when something is changed, well, their behavior immediately change, and then it adapts to in a kind of homeostatic way. So, for example, if you change the medium, remember that it's a single cell. So if you change the medium, they're basically directly in contact with it. So it's going to affect them tremendously, and so it does affect them. But then the properties of their ionic channels change, and the behavior uh, comes back to normal, but in a different context. And in the same way, if you culture them at a different temperature, well, they're going then to like that new temperature. And in addition to that, physiologically, they are going to change and adapt to it. And finally, there are also cases of learning. Not many cases in the literature, but they are there. One is tube escape. So tube escape is well, it's a simple behavior where you put a capillary inside the dish, and so they are brought into the capillary, and because they do gravitaxis, they go upward, and then they are kind of in a dead end. They bump into the surface of the meniscus. Basically, they are trapped. But after something like 30 seconds, they dive in and they escape. Now, if you do it again, they will escape in 15 seconds. So, it seems that they have learned to escape the tube. It's not very well understood, but the behavior is has been reproduced a number of times actually. Um, Another known case is conditioning, associative conditioning. So this is a paper from 1979 where they show that they can uh, train a paramecium to respond to sound. Normally, if you produce sound, Paramecium will, well, it seems that it doesn't hear anything. Nothing happens. So you can see here with the 500 hertz tone, you can see a trajectory here. It's not affected by the tone, but if you couple it with an electrical shock, then after just a few pairings, it will respond to the sound itself, just the sound without the electrical pairing. So that's pretty interesting, and it has the properties of conditioning that is seen in other species actually. So
Sorry, Roman, there is a question in the
sure.
just for like something you showed previously. Mike is asking how different is the paramecium direction change from the E. coli run and tumble?
Okay, that's a good question. So there is some similarity with run and tumble in bacteria, and also some differences. So um, the first thing is that paramecium does not only change direction, it also swims backward for a little bit. And the second big difference is that the tumble in bacteria is seems to be really random, whereas in Paramission it's not really random. It's they always move um, with they always turn in fact with the mouth facing the axis of the rotation axis, so they always move in this kind of the same direction, but it's graded. And so it's a bit less random. It's also graded, that is, it depends on the stimulus. And so these differences in stochasticity in particular are, well, they are probably due to differences in scales, because paramecium is about 100 micrometer long, whereas bacteria is maybe a few micrometer. So so in terms, so you have much more noise on the scale of bacterium than on the scale of paramecium, because you have well, 2 orders of magnitude difference for the scale, so that's 4 orders for the membrane. So it's a lot of difference. So basically, you expect the noise to be 100 times larger in bacteria. Uh, so there are a few differences, but also some similarities. For example, it's known that chemotaxis in bacteria uses this kind of trial and error also uh, behavior. That is, they do the tumble when they're not going in the right direction, except it's more stochastic, basically. Does that answer your question?
Yes.
Okay, good. Um, all right. So now I move on to the modeling. So, so what we wanted to do is a model of the action potential, like the Hodgkin-Huxley model, basically, but a model that is coupled to the motility, so that we actually get a model of the swimming cell. So to do that, well, we have developed a little device to immobilize the cells to do electrophysiology. So it's a simple microfluidic device, which is basically a filter. So you put the cells on a filter, and then from below, you pull the fluid and put it back, and so the cells are basically stuck to the filter. And then, as you can see, we can put two pipettes, two electrodes there, and we can send pulses, current pulses, and record responses. So here you see a, well, a tiny action potential with a small current pulse. And at the same time, we can do some videos. So it's not so easy to see the cilia moving because they go quite fast. So what we have done instead is to measure the motion of the fluid. So we put uh, small uh, particles of silica in the fluid, and so they move with the motion of the fluid, and we image that. We can calculate the motion field, which I show here, and from that we know in which direction the cell is trying to swim, basically. So here you can see that before the stimulus, it's trying, the cilia are beating, also the fluid is moving this way. So it's trying to swim forward. And then during the stimulation, you can see that fluid motion reverses. It's trying to swim backward. And then it reverts, and then actually the fluid kind of whirls around the cell, and it's going to turn. And that corresponds more or less to what you see in an avoiding reaction with the backward swimming and then turning and then forward again. So that's basically our experimental setup. And what we have done is basically some model fitting that includes both the electrophysiology and the motion of the fluid, of the cilia, basically. And so here it is. Here are some responses that are measured. These are responses to pulses. And so you have graded responses. Um, you can see here, for example, that there's an hyperpolarization. So that is due to a calcium-dependent potassium current. So as I mentioned, at the same time, we image fluid motion. So when we have the fluid motion, we calculate the mean angle of the motion, and that's shown here. And so we can see that it reverses. So basically, we can measure the reversal of the cilia in this way, and it actually depends on the strength of the stimulus. On the right, you have a zoom on one of the responses, which shows a small action potential. And if you, from this response, you can calculate the transmembrane current. And you can see an inward current, and that corresponds to the calcium that is getting in. And then you have a potassium current, which is the delayed rectifier current, which you find in other in neurons, basically. Um, Then if you integrate the inward part, then you can calculate basically all the charge that gets into the cilia and deduce how much calcium gets into the cilia. And if you do that, you find here that you have an increase of about 10 micromolar. So to give you an idea, uh, in the resting state, the calcium concentration is something like zero point 0.1 micromolar in most cells. So there's a large, basically a large influx of calcium, and many calcium binding proteins in the cell bind around a few micromolar. So that's what happens here. Now we have tried to fit a model to this data. So the model was obtained, um, well, first from the electrophysiology literature on paramecium. There's actually a lot of literature, so we know basically what kind of currents are there, and we know the basic properties.
Is it
So
known how many calcium channels
okay,
there,
so
there is? That different
yeah,
types?
yes, so more or less. So the genome has been sequenced, so we have all that information that we can search, and so they are, um, well, at least as many channels as we have, basically, in our neurons. Many potassium channels in particular. You have calcium channels, also sodium, but maybe calcium-dependent sodium channels. All sorts of channels that you find in neurons. Many of them you can find in paramecium. So I would say there are a few hundred genes for channels. Uh, it's hard to say exactly how many types of channels that make, because there are some subtleties in the genome of paramecium. There are duplications of the genome. So um, you have whole genome duplication in the genome for some reason. So you have yeah, maybe two or three duplications of the genome. So you have genes that are copied at different places, and then with time they have some differences. So in the end, it's a bit complicated, but let's say it's a few hundred genes. But what we have modeled, of course, is not a few hundred channels. It's essentially three channels. It's so let me see. I can see it. Yeah, I have to move the zoom window. All right. So this is basically the Hodgkin-Oxley model that you see. So you have the membrane equation, and then you have three channels plus the leak current. The three channels are, well, the delayed rectifier current, which is the same as you find in squid axon, so in the Hodgkin-Oxley model, um, except with different parameters and so on. Uh, then you have the calcium channel. So that basically plays the role of the sodium channel in the Hodgkin-Huxley model, with one important difference, which is that the sodium channel in the Hodgkin-Huxley model inactivates with voltage, but this one inactivates with calcium. So you have a kind of, you have a negative feedback loop in this way. Calcium gets in and inactivates the channel. So this is the second equation here. And then you also have a potassium current that depends on the calcium. So this is basically illustrated here on the left. You have calcium that enters the cilia through uh, the calcium channels, and then the calcium is going to feed back on the channel and inactivate it. So this way you have just a transient current, and then it's going to activate a potassium current, and of course it's going to activate the motors, so that the cilia are going to move in a different direction. So this we simply model with a function that relates the calcium concentration with the angle of the cilia. And then the calcium is going to be expelled. So you have PMCA, so that's calcium pumps that you also find in all species, basically. And there are some calcium pumps in the cilia. So there's an equation here which is the calcium dynamics, basically. So that's the main things. So we have a number of parameters, and we're going to do model fitting of those parameters.
There's another question in the chat.
Sure.
Aspar is asking if does the gene duplication affect transfection studies, which is like the expression of exogenous membrane channels in that organisms. The fact that there is more than one set of
Sorry,
genes.
but I'm not. I don't know much about genetics, so can you be more precise? What do you mean exactly?
Just go on, Espoir, or can you just...
Yeah, sure. You mentioned that this is a great talk, by the way. You
Thanks.
mentioned that the genome duplication for the
Yeah.
species, there's multiple duplications. So if you were trying to titrate, for example, expression of exogenous
I see.
rhodopsin, could you do that? And can you calculate can you predict how much channel rhodopsin you can express, for example, if you were trying to control for that?
I have no
Theoretical experiment.
I have no idea because it's not really my expertise, I have to say. So I don't know how you do that. What I can say is
I think
that
Andy Cohen might have. I think Andy Cohen might have optogenetic paramecium, but I don't remember.
Optogenetic? Seriously?
I think so.
Okay, it'd be interesting to know because I've never seen that in the literature. So, okay.
Thank you.
Let me know. Let me know. Yeah, I'd be interested to hear about that if you.
Ian, just feel free to ask question directly, like if you want to during the talk.
Okay. Should I move on or?
Yeah, sure.
Yeah. Okay. Okay. Um, okay. So we have this model that we are fitting to the data. So Frederick mentioned Brian. So sorry, every time I'm trying to move the video because it masks my slides. Anyway, right. You see this? So we have developed a model fitting toolbox with Brian, which is available online, so you can get it. And basically, you put the equations of your model, you give your data, and it fits the parameters, and it does that with a combination of a um, population-based optimization. So you have, well, basically it uses several existing packages. One is the Facebook optimization package. So it uses that and also gradient descent. So because from the equations we can calculate symbolically, so we calculate symbolically the gradients, and we do gradient descent. So it's quite efficient. So that's what I used to do the model fittings, and you can see the results of the model fitting on the right. So, and it works okay. And so this model fitting also gives us access to hidden variables that are in the models, such as, well, the different currents, the different tuning currents. You can see here, for example, the calcium current, which is transient, and then potassium current. And you can see also the calcium concentration in the cilia. So you can see here calcium concentration increasing upon stimulation and then more slowly decaying depending on stimulus strength. And the dashed lines here, I think the first dash line, is it the first dash line? Yeah, the first dash line is the threshold basically for ciliary reversal. And that comes also from the fitting. And turns out it corresponds, and we're quite lucky, it corresponds to what has been measured directly. This has been measured, yeah, in different ways I can explain if you're interested. Um, so, okay, so now we have basically the model of the action potential coupled with ciliary reversal, but that doesn't give us swimming cells yet. So we have to add basically the kinematics. So we can measure the direction of cilia beating. When it's moving forward, it tends to, well, so the cilia beat backward and a little bit to the right. And when it swims backward, the cilia beat a little bit to the right. And what that produces is a helicoidal movement, like it's screwing basically. So this is an example. It's kind of screwing itself in the water. And you can see maybe a little bit here, this is the oral groove. And the oral groove always faces the axis of the spiral. So the kinematics can be parameterized by these parameters, basically. So we simplify the kinematics a little bit. You have the translation, which is in the direction of the main axis. So it's parameterized by velocity, which can be negative. And then the rotation axis makes an angle with respect to this main axis towards away from the mouth. So there's an angle theta, and then it spins around this axis by some spinning speed omega. So we have three kinetic parameters that determine the kinetics. Okay, so now how does it turn? So when it swims, it's kind of screwing itself in the water. But how does it change direction? So for it to change direction, what needs to happen is that the rotation axis must change at some point. And so that is basically what is described in the literature by Jennings. You can see examples of different graded avoiding reactions where the angle increases. And so basically during the avoiding reaction, the angle is going to increase a little bit. And so the cell is going to turn, but in a new direction. And then the rotation axis goes back to normal and it moves forward, but now it's in a different direction. And so to understand what's going on, we can look at hydrodynamics in a spherical model because you can do analytics there. And this is this is what I've done here. You can calculate basically the torque and the motion of the cell for a given pattern of ciliary beating. So if you have a pattern which is homogeneous around the main axis, you're going to have well, a motion that is going to be along that axis and also turning around that axis. The rotation axis is going to be along that axis by symmetry. And if the cilia beat in the reverse direction, well, you will still have rotation along the main axis. So you cannot have turning in this way. To have turning, you need to have some cilia beating in some direction and some other cilia beating in another direction. And this way, the rotation axis is going to be different. So in this picture, the rotation axis would be kind of orthogonal to the slide, okay, if you want. So, and this you can calculate. And so you can calculate in this configuration, for example, that the angle theta will be around 30 degrees in this case. So this is what we think is happening. During the avoiding reaction, some cilia will be reverted and others not. And this will happen if there is an inhomogeneity in the ciliary reversal threshold of the different cilia. So for example, if you look here, the cilia that are on the left top, they have already reverted, whereas the one on the right have not reverted yet. So if they have different calcium threshold for reversal, you will have this transient inhomogeneity that will result in turning in a different on a different axis. So ideally, we would want to measure the ciliary beating pattern during this avoiding reaction all across the cell, but that is very difficult to measure. So what we've done simply is to give some evidence that this happens. So there's some evidence in the literature. What you see here is electron microscopy that is done during avoiding reactions. And you can see that you can see inhomogeneous patterns here. So what you see here is the so-called metachronal patterns. That is the waves, basically, that the cilia do. And so they do a wave that looks like that during forward swimming, but during backward swimming, they look like that. They have a different angle. But when they revert back, well, the posterior part, the posterior part reverts later than the anterior part. So that's what the electron microscopy suggests here. And we have basically confirmed this by imaging the motion of the fluid. So you can look at the time of reversal of the fluid in the anterior part and the posterior part. And basically it happens late in the posterior part. But ideally, what we would like to show is that you also have inhomogeneities within the anterior part between different sides. But for this, we would need different techniques, basically, maybe directly imaging cilia motion. So we just have some evidence of this. And what we're going to do now for the model is to use a phenomenological model, which couples calcium concentration to these three kinematic parameters that I've mentioned, which are the translational velocity, the angle of the rotation axis, and the spinning speed. And what we've seen is that, so for the velocity, above some threshold, which is in the micromolar range, the cell is going to move backward. So the function that relates velocity to calcium concentration will look like that, so a bit of a sigmoidal function. And the two other, the rotation parameters, variables, what we've seen is that they are going to increase basically when the cilia revert. So it's going to be around the calcium threshold for reversal. So in the micromolar range, you will have an increase in theta, in the angle, and in the spinning speed. It should look like that. Now, the numbers here are kind of, well, they're not direct measurements. Well, so a few points here are direct measurements. So basically what we can measure is the angle during the forward swimming, because we can just look at the spiral. And we can also have the spinning speed during the spiral. So these 2 points are measured. Then the maximum here is 90 degrees because that's what corresponds to the largest avoiding reaction that are illustrated in Jennings' books, basically. And for the spinning speed, it's just the factor that we've seen in the spherical model. So these are a bit rough models, let's say, phenomenological model, but they give us a complete kinematic description of the swimming cell that we can now integrate. So basically, we add this to the electrophysiological model, and then we integrate the kinematic variables, and we have a swimming cell. So here on the left, you have a simulation of the swimming cell. It's doing spirals, and then we send some short pulses at different times. And so you can see that with a very weak pulse, you have a change in direction without backward swimming. And with a strong pulse, you have a small backward swimming followed by change in direction. On the right, you see an example that is a measurement of an actual cell that is swimming that does two avoiding reactions. Well, one is not exactly an avoiding reaction, just a change in direction, and then an avoiding reaction with backward swimming, which looks a bit like our model. Okay, and now the movies. All right, so now we can simulate the model with a simple environment. So now what I've done is that I've considered a half plane. Basically, the stimulus is a half plane. That's the object. It's an actual object in the sense that there's no mechanical interaction. So let's say it could be a chemical region, and I model the stimulus as um, giving a current to the cell that is proportional to the surface of the membrane that is in contact with the stimulus. And so this is what happens. Okay, that's the simulation. Okay, so what you see here, lots of small avoiding reactions. So if you model it in this way, what you can see is that it's doing very small avoiding reactions, and this makes sense because now you don't have simply a stimulus response that like we usually do in neuroscience. You really have a closed loop. So the stimulus here actually depends on the behavior, on the reaction of the cell to the stimulus. And since it's going backward as soon as it enters the stimulus, you will have a very small stimulus. And this is what you see here. So here you see the membrane potential. Um, in gray is when it enters the gray zone. And in blue, it's when it starts swinging backward. So basically, very quickly, it starts swinging backward and then it exits the stimulus. And so you have a very small stimulus. If you want to have an avoiding reaction, this means that you need to, in fact, change the transduction model so that you have a longer stimulation. And so that's what happens if you simply have a slower transduction. So here I have a transduction model which has the time constant of a few tenths of milliseconds. And now this is what happens with exactly the same model is you have a stronger avoiding reaction with an avoiding reaction which now lasts 150 milliseconds. Okay, so now we can simulate the model in an environment that is a little bit more interesting. So now what I'm going to show you is, I think, something like 100 cells, which are simulated with Brian, by the way. So it's simulated with Brian, both the kinematics and the action potentials and everything. And all right, so now the stimulus is a disk, and the environment is actually a torus. So that when it goes to one edge, it ends up in the other edge. And so you can see that they're doing avoiding reaction at the edges here. So this stimulus acts as a repelling stimulus. You can see that sometimes cell is not very clever because sometimes it does an avoiding reaction, turns, but does a very large turn so that it ends up being backward into the stimulus and then swings backward until it exits the disk. Right, now how can you have an attractive stimulus? So I showed an attractive drop of weak acid that was illustrated by Jennings. And how do you get that? Well, to get that, you need to have an avoiding reaction at the edge of the stimulus, but only once it's already inside the stimulus, but it has to enter it. And so how do you do this? Well, a simple, basically what you want to have is sort of rebound when it exits the stimulus. And this you can get with adaptation, for example. So if you have a stimulus which is hyperpolarizing, so when it enters it, it's going to hyperpolarize the membrane, so it will just enter and nothing will happen. And when it exits the when it exits the stimulus, the membrane potential will return to normal, but you want to rebound. So this you can have, for example, either with adaptation, or here what I did is even simpler than that. You consider that the stimulus actually triggers two currents, one that is hyperpolarizing and fast, one that is depolarizing and slow. And when you add the two currents, you have basically a transient current, which is hyperpolarizing when it enters and depolarizing when it exits. And if you do this, then you find that the cell um, will kind of bounce on the edge, on the inner side of the stimulus. I don't know why the movie is a little bit bouncy like that, but that's just the movie. I don't know. Anyway, and so now the disk is attractive. Now, if you take exactly the same model, but now instead of having a disk, use a linear gradient of stimulus. So let's say it's the concentration of some chemical substance. Then this is what happens now. So someone mentioned run and tumble, and this is basically what's happening here. The cell is going to do an avoiding reaction when it's moving in the wrong direction. And the result of this is chemotaxis is going to move in the direction of the increasing gradient of the substance. So you can see here, you have a few hundred cells that are following this gradient. And it's the model is still the same. I haven't changed the model actually. I just changed the environment, just the stimulus. Now you can do this again. Change the stimulus. So the next thing I'm going to show you is the same thing. The paramecium are in an environment where there's nothing in the environment except that they breathe. And so they breathe, they produce CO2. And the CO2 makes the fluid acid, and they like acidity. So what I'm going to add here is just the production of CO2 by cells and diffusion of CO2. And if you do this, so now I'm representing the cells, and the background is the concentration of CO2 normalized. And so you see that's some inhomogeneities, and these inhomogeneities are together with this chemosensitivity of the cell is going to result in collective behavior. This is basically a reaction diffusion process that you're seeing here. Now, after some time, I might have to move forward a little bit, but you can already kind of guess what's happening here. They are starting to gather, and because they gather, they produce more CO2. So it's going to amplify the phenomenon, and after some time, you can see these things happening. They're going to gather in some places. All right, so this is basically all I wanted to show. So, okay, just to mention what we are working on now, a lot of empirical work, actually, that we're trying to do. So I mentioned that parts of the model are phenomenological or not completely empirical, well, or at least a bit rough. In particular for the motion, how it turns, it's not so easy. So we're trying to do some measurements in 3D and to see some details of how it turns. Another part of the empirical work is to model the electrophysiology, but for sensory transduction, so chemical or mechanical. So we've done some mechanical work now. We think we've identified the mechanoreceptors. And some theoretical work. So now it's theoretically quite interesting because we have a model which is not just a model of the action potential that the Hodgkin-Oxter model, but an actual model of an organism that swims. So we can study it from a dynamical systems point of view, but now it's not just the electrical behavior, it's actually the behavior of the cell. So we can look at different interesting questions like, well, how does it avoid obstacles and things like that, and how that can be expressed in terms of, dynamical properties, bifurcations, and so on. And the third thing that we are trying to work on is to look at well, more complex behavior like learning and adaptation. So that's basically it. And I just wanted to mention a number of people I'm working with on this topic. So mainly I'm working with another lab of physicists, the lab of Alexis Préau and Laetitia Pontani, also in Paris. So it's a biophysics lab. They're working in particular on mechanical biology. And they have done a lot of work basically on the behavior. So you can see a video here on a paramecium that is swimming in a little well. So what they do in particular is make some swimming pools with obstacles, and we're trying to understand how they avoid obstacles and so on. And so Anirudh Kulkarni and Irene Elises are the ones who did the electrophysiology. They were postdocs. Irene is still a postdoc. Nicola is a PhD student who's doing behavior. And Eloise is currently doing some 3D behavior with several cameras. And also, we're working with the specialist of paramecium genetics, who is Eric Meyer in INS in Paris. So we're trying in particular to, well, identify the mechanoreceptors and also to develop a calcium indicator, a genetic indicator. So that's the work in progress now. That's it. Thank you for your attention, and any question, happy to answer.
Thank you for like nice collaborative work on the Paramission. I hope
Thank you.
it will stimulate people for doing more collaborative work. So please feel free. We have like perhaps 10 minutes for questions. So feel free to ask Romain directly the questions, or write them on the chat if you feel, and I will read it to Romain.
Yeah. Okay, so I need to see how.
Do
to
you
put
know?
this in.
Do you know if E. coli also does paired stimulus learning?
If sorry, I didn't hear the
If E.
beginning.
coli also does paired stimulus learning?
Conditional learning. I've never seen that, but I'm not the expert on that.
Yeah.
But yeah,
Okay.
honestly, I don't know. Yeah.
This is fantastic work.
Thank you.
Oh, sorry. I was gonna just follow up and ask whether the calcium sensitivity can be manipulated if you wanted to see that model convergence for detection, the chemotaxis. If you wanted that to convert faster, for instance, could you change the calcium sensitivity of the organism?
I'm not sure I got it. Calcium. What do you mean by calcium sensitivity? So there.
I'm sorry, the CO2 sensitivity.
Okay. Can it be manipulated? What do you mean manipulated?
Could one increase the sensitivity for CO2 such that they respond faster, for instance, to a change in CO2?
That's a good question. I have no idea. So the sensitivity to CO2 is probably mediated by acid sensitivity. So I guess this might be modulated, but we have not identified the receptors for that. If we do, then I mean there are known receptors for acid sensors, which are known to be modulated by various things. So it's very possible. I mean, I would expect that chemical sensitivity is probably modulated by metabolic state, like if they're hungry and so on, or if they're feeding and things like that. So I would expect that, but it's not known as far as I know. That would be quite interesting, I think, to do that. But maybe the first step might be to identify the sensors, which we could do, I think, because there are many things that we can do with paramecium. That's one advantage of this organism, is that we can do genetics quite easily. So we can do a RNA interference, for example. We can knock down a gene by making them eat bacteria that express some plasmids, that have some plasmids. So that's things that we're doing for the mechanoreceptors now, for example. So I guess we could do that for the acid sensors too. So that would be a way, I guess, to look at this question. But I don't know the answer to your question for the moment.
Thank you so much. I really appreciate the answer. Thank you.
Thank you.
I was going to ask for the attractive stimulus, you made two mechanisms. You said one could be through adaptation, or the other could be with the two different speed currents, and you chose to use the two different speed currents in the model.
Okay.
I was just wondering which one you think is really going on.
So first, the reason I chose this, actually, initially I tried something else, but this one was just simpler. So it was more kind of proof of concept, because the way I modeled it was not specific to a particular kind of stimulus. Because there can be adaptation of the sensors themselves. So for example, in bacterial chemosensitivity, for example, we know that it is the chemical sensors themselves that adapt. So I think they are phosphorylated or something like that. And so they adapt. And there are models for that. But you can also have adaptation of excitability, for example. So this we know also exists. So the different things can be going on, basically. So so that's why I chose just the simpler mechanisms that just show the principle. But I think you can have either modality-specific adaptation, or you can have also adaptation of excitability, which would then apply to all modalities at the same time. So and there's evidence of the second type for sure. Yeah, but for this, I think we need more experiments, because we haven't done any experiment to get the models of transduction yet. In the literature, there are just a few papers where they measured responses to a few chemicals, but not so many actually. And mostly in the literature, there are studies on mechanotransduction. And for mechanotransduction, there is adaptation. That is for sure.
I guess
that's all I can tell you.
What is the time scale maybe for this adaptation and for the conditioned response that you mentioned? About how long do those memories last?
Okay, so for adaptation, that really depends on the stimulus. So I've mentioned several things. So there is fast adaptation that has to be involved in attraction by acid stuff, for example, because this has to be on the order of seconds or a few tens of seconds maximum. But then there's adaptation that I mentioned before, to changes in the environment. And this is rather a couple of hours. Okay, and then there's mechanical adaptation. So for example, if you stimulate the cell mechanically, like if you shake the tube or something like that, you will see that they swim faster for a few minutes. So you really have different time scales. Then for conditioning, you mentioned that's what you wanted to know. Conditioning, it's pretty fast. So conditioning, the experiments, the trials, they last just a few seconds. And after just a few trials, they see the conditioning. So it's really some seconds. But then there is retention, and that might take longer. This I don't remember exactly. But to see the phenomenon, it's just a few seconds actually. It's been quite fast, this conditioning. Yeah.
That was great. Thank you.
Oh, I'm sorry. I wanted to follow up on that. I don't know if I heard the answer, but how long does the memory last? How long can you sustain that?
Yeah. Okay.
Conditioning?
Okay, yes, it's a complicated question actually. So the conditioning, if I remember correctly, you would have to check the paper actually, but um, I think they show retention for a few hours if I'm correct. Then there are also people have seen retention of some. It's not exactly conditioning, but changes in behavior due to feeding and stuff like that, which can last more than that. But then if you go beyond a few hours, you get a reproduction because the cells, they can reproduce every 6 hours in the right conditions by fission. So they divide in two. And if they divide in two, well, you can have retention of the memory in the two cells in principle. So this there it gets a bit more complicated because you have different new cells. So yeah. So then how much time when you have division can you retain a memory? This is an open question, I guess. Yeah.
Thank you.
I have a question. When the paramecium runs into an obstacle and then turns and changes direction and moves away, one end of the paramecium detected the obstacle, but the whole animal
Okay,
had,
that's okay.
the whole animal had to change direction on the cilia in order to get movement. There's not a nervous system in there to cause it to change direction. How could this be done with distributed kind of control?
Yeah, not exactly sure, but okay. All right. So for this change in the direction in the mechanical aspect, actually there are different receptors in the anterior, in the front, and in the rear. So in fact, if you touch it in the front, it will swim backward and then change direction. But if you touch it on the rear, it will move forward. And this happens because there are two different kinds of receptors. So in the front, you have depolarizing receptors, and in the back, you have hyperpolarizing receptors, and then they trigger different responses. Then the change of direction itself is basically random. It does not really depend well, on anything. So what's going to happen when it hits an obstacle is going to move backward and then basically try a new random direction and then move forward again. And maybe it will hit the obstacle again and change direction until it escapes. So it's more an exploration kind of mode of control.
I understand.
Trial and error, basically.
Okay. I think I understand that, but what I'm getting at is all the cilia kind of have to agree on what direction they're going to change to. And their sensor is only on one end when they're bumped.
Yeah, so that's not exactly what happens. So the way it changes direction is not by the cilia going into that direction. It's by cilia, like a bit like in the tumble of bacteria. The cilia are a bit in different directions, and that makes the cell move kind of not randomly, but in a new direction, turn in a different axis. So it turns in a different axis and then it stops. And when it stops, it is in a different direction from before. So the cilia don't choose any direction. They choose to bit in an asynchronous manner so that the cell turns. Okay. It does not choose a direction.
Okay. I think I see.
Yeah.
Yeah. It just kind of randomly gets a new direction and then they all just start moving forward again.
Exactly. Yes, that's right.
Okay.
Yeah.
All right. Thank you.
Is this, sorry, one last question. Is this through calcium transient? Is this through intracellular calcium transient? How do you, how do you coordinate this movement?
Yeah. So, so as I said, the action potential is calcium based. So the calcium channels, they are located in the cilia, not in the main membrane. They're in the cilia. So when they open, the calcium enter the cilia, and there, well, the calcium binds to a bunch of things, including some motor proteins, and that makes well the cilia change direction basically. I don't know the details actually of this molecularly, but they bind to some stuff, the motors in the cilia, and yeah, through the calcium transient. That's the main messenger. There's also cyclic AMP that is involved in this, so it's not as well understood. But
I see. So
yeah.
theoretically, you could transfect like GChem, for instance, calcium dye indicator, and actually visualize this.
Yeah, that's right.
Coordination.
So, yeah, in principle. So some people have done that a little bit for, I mean, in Paramecium, people have done some calcium imaging, but on kind of long time scale. They were not looking at the action potential, but actually at another thing I mentioned, the trichocyst ejection. So there has been some work on calcium imaging with the micro-injected calcium indicators. And so this we've done also, and but it's a bit difficult to do actually, because ideally we want to see a calcium change in the cilia, but that's a really small volume compared to the whole volume of the cell. So the signal is small, so it's not so easy. So this is one thing that I mentioned at the end. We want to do a calcium indicator that is genetically encoded and that is targeted to the cilia. And in this way, we could see really the calcium in the cilia specifically.
That's right, exactly.
So I don't know if we'll manage to do that, but that's
wonderful.
Yeah. Yeah.
How much more do you think you'd have to add to your model in order to study something like prey capture?
Something like?
Prey capture. So hunting.
Okay, well, prey capture. Well, I mean, phenomenologically, it's not very difficult to add because what they do is so they have an oral groove. The way they feed is that they pull bacteria, for example, they pull the fluid into their oral groove. So they have very powerful cilia along their oral groove, and so when they feed, they basically, well, typically they attach to some fiber. And then the cilia, they bring whatever is around into their oral groove. And so
no, I meant like ecological scale. So like you have that reaction diffusion equation.
You mean like doing chemotaxis and this kind of stuff?
Or just like, so you have like, you have this eco scale where you have
I see.
like.
Okay, well, I guess there are, well, what you want to have is to model an environment, so maybe with bacteria or this kind of stuff. Well, this I guess is not that complicated if they can move, and then signals. So, well, that depends what kind of. I mean, it's there's really no end to this, but at least you want to have, well, production of some metabolites, or maybe CO2 or something, but by bacteria, and some diffusion. So that's, well, that's almost what I showed you in the last slide. Right. Then maybe you want to have some chemical sensitivity. And yeah, if you want to be a bit realistic, you would want to have CO2 sensitivity. But yeah, well, no, actually, you would also want to have some mechanical aspects, I guess. Because you want them, for example, to attach to something and feed there, this kind of stuff. I guess the feeding behavior, that's called thigmotaxis, actually. When they attach, they attach to a fiber, they feed, and then they leave. And I'm not sure how that happens exactly. So this is maybe one thing that would be good to add to the model. But we're not very far to, I mean, it's not a lot of things to add to the model in terms of equations, I guess. Maybe some mechanics we want to have here. Yeah, to avoid the mechanical part, what I did in what I showed you is to have environments that have a torus topology. So there are no boundaries. And of course, that simplifies the modeling quite a bit. But if you want to have a real environment, then there will be the surface of the water and this kind of things. And so you would need to have mechanical interaction with surfaces, basically. This is one thing that should be added, I think. Unless you want to keep a torus topology, but maybe, well, yeah, it's less realistic, but yeah. Does that answer the question? Or?
Oh, yes, yes.
Okay.
All right. Thank you, Romain.
Thank you.
It was a very nice talk.
Thank you.
Good luck with your experimental work.
Yeah. It's quite fun. Yeah.
Might hear you next year for like the second part.
Then yeah, okay, sure. Okay, thank you. Yeah, the paper, by the way, is on bioRxiv if you want to check that.
Okay.
Yeah, well, if you go to my website, you'll find a link to it. And there's the movies and the code also and the data. Everything's there. So if you want to have a look. Yeah. Okay. Well, thank you.
This was a lot of fun. Thanks.
Thanks.
Thank you.
Yeah,
Bye.
interesting, very much. Thank you.
Thanks. Okay. Well, so see you another time.
All right. Bye-bye.
Bye-bye.